Tonight's event is the second lecture in a series dedicated to the memory of Christine Mona Khademi. A generous gift by Christine's father, Monib Khademi, established this annual lecture in her memory to support and highlight the work of women in particular who are making substantial contributions to Iranian culture and society in the fields of politics, science, technology, public service, art, or culture. We are so pleased to have Mr. Khademi as well as Christine's mother, Julia, and family and friends with us here today. In addition to the annual lecture, Mr. Khademi's gift supports Stanford undergraduate and graduate students conducting research related to Iran with a major focus on women and their impact in social, scientific, or political arenas, research that promotes finding common ground and encourages tolerance in Iranian society, or programs designed to empower and integrate individuals with neurodivergent thinking or communication challenges. This is new, and for our Stanford students in the audience, I really encourage you to take a look at our website and read the details. It's a fantastic opportunity I hope you'll take advantage of, and you can find out all about it on our website. Again, join our email list, and we're sending out details. Christine Mona Khademi graduated from Stanford with a BS in biology with honors in 2012. And by all accounts, she was kind, compassionate, and inclusive. She aspired to be a doctor. Christine sadly left this world too soon, and I regret not having the opportunity to have met her while she was at Stanford. I really encourage you to read the full biography that we have printed tonight to learn more about the remarkable young woman she was. We are honored to be a part of celebrating her memory tonight and every year, thanks to this gift. This annual lecture series launched last spring, and I see a lot of familiar faces from there, with a fascinating inaugural talk by Dr. Mariam Shonechi from the University of Southern California on the intersection of AI and neurotechnology. That lecture is on our YouTube channel for anyone who'd like to go back and watch it. Tonight, we are so pleased to be hosting Dr. Nikta Fakhri to carry on this tradition. She is an associate professor in the Department of Physics at MIT and the Physics of Living Systems group. She completed her undergraduate degree at Sharif University of Technology in Iran and her PhD at Rice University in Houston, Texas. She was a Human Frontier Science Program postdoctoral fellow in Germany before joining MIT. She is an Alfred P. Sloan Research Fellow in Physics, and she is the recipient of numerous awards, including the 2018 International Union of Pure and Applied Physics Young Scientist Prize in Biological Physics, the 2019 National Science Foundation Career Award, and the 2022 American Physical Society Early Career Award in Soft Matter Research. This is just a small part of her extensive bio, so we're delighted to have her tonight to lecture on the hidden order of life. But before welcoming Dr. Fakhri, we invite you, having battery issues. We would like to invite Mr. Khademi to say a few words. As a Stanford alum himself, we are grateful to him and his family for their ongoing support of academic programming at Stanford Iranian Studies to promote peace, democracy, and inclusivity in Iran and around the world. So please welcome first Mr. Monib Khademi.
Thank you very much. خیلی متشکرم از دوستان عزیزی که اینجا هستین و دوستانی که این برنامه رو توی اینترنت در بعداً خواهند دید. با اجازهتون I'm going to just speak in English. So thank you for being here. Christine was a brilliant young scholar who spent some of her happiest moments of life here at Stanford. I remember her excitement when she was first named in a published scientific paper. She sent me email, she called, she said, "Dad, I'm published." This evening, as we gather to celebrate Christine's legacy, it's important to reflect on the impact she had on those around her. Her passion for learning, her unyielding curiosity, and her dedication to making the world a better place were qualities that defined her. I remember her after school, in high school, she would go to our market, Mill Valley Market, and solicit money in coins and small bills for books for children in Afghanistan, for people who are hungry in Sudan and other occasions. Christine's journey was marked by a relentless pursuit of knowledge and a deep-seated love for her heritage and roots. She was also funny in her own mischievous way. Tonight we are not only remembering her, but we are looking forward to the future she and many in her generation have envisioned. A future where Iranian culture and contributions, we hope, will continue, and we celebrate that globally. Through these lectures and projects, we hope to continue her vision to inspire future generations and create a platform where voices like Christine's can be heard and celebrated. This evening, I also acknowledge the broader context of our current world. Amidst the chaos, challenges, and disruptions, it is moments like these that remind us of the resilience, toughness, and depth of human spirit. The relentless pursuit of excellence, the courage to innovate, and the power of education are what bind us together as we navigate these days in our lives. After all, we live one short life on this small planet, a magical planet of ours in this vast universe filled with billions of galaxies. Me and my family are also very grateful for Stanford continuing to lead, innovate, and maintain a world-class institution here in the Bay Area, where most of us live. I thank Dr. Abbas Milani, a great teacher, a very special friend, who I've now known for decades. You've created an incredible home for the Iranian culture and community that is alive and thriving with its unique diversity. Our three daughters, Christine, Courtney, and Casey, all took classes at Stanford from Dr. Milani. They had memorable time in their studies with you, as you know. We continue to honor and cherish you, Abbas, so that you know we are very grateful to you. And Roma, we also know that without you, the various offerings of this program would not have been possible. So thank you so much for your meticulous leadership and for choosing to make your contributions here in this particular way. We are grateful to you. My family and I expressed our deepest gratitudes to Professor Nikta Fakhri and all you esteemed guests who join us tonight. I first heard Dr. Fakhri speak a few years ago, and then I called her at MIT, and she was gracious to answer my phone, and we talked, and I found a few points about her amazing life. In our own small way, we honor you, and we honor your family, your sister, and in particular, we also, who's a professor here at Stanford Medical School, and in particular, we honor your parents, who are also here tonight. And your courage in both raising your children and the courage to immigrate to this country. After all, U.S. continues to only be an evolving experiment in democracy. So we hope that that continues and we learn from developments in our country. Your presence here, Dr. Par, is a testament to the enduring bond between cultures and our shared commitment to fostering knowledge and understanding. Let us continue to champion the values Christine held dear. Let us strive to build bridges, break barriers, and create a world where every individual contribution is recognized and valued. In closing, may also honor Zan Zendegi and Azadi, Women, Life, and Liberty, which continues to ring true not just in Iran, but here in the US and around the world. Thank you all for being part of this special evening and for helping us keep Christine's spirit alive through this meaningful celebration. Thank you very much again.
Thank you, Monib, and the entire Khademi family for this honor and privilege to be here to celebrate the life of Christine Mona Khademi, and also to Hamid and Christine Moghadam Program in Iranian Studies for organizing this event, and to all of you for being here. I can think of no better way to celebrate life than to talk about life. Now, imagine a crisp fall evening, the sky a canvas of amber and violet. Suddenly, thousands of starlings appear, performing a breathtaking aerial ballet. This ballet is called a murmuration. It's a stunning natural phenomenon that holds the secret to the very essence of life. I am a physicist, and I see something fundamental in flying of these birds. I see fundamental principles of self-organization that govern not just these birds, but all of life, from the tiniest of cells to the vastest ecosystems. So where do we go from here? Let me take you on a journey to discover the physics of non-equilibrium systems, like these starlings, that can account for the phenomena of life. Now, as a biophysicist, I am fascinated by the fact that there is something different about being alive, and I would like to know what that is, in the same way that I know what it is for a collection of atoms to be solid and for a collection of electrons to be superconducting. Now, if you look around, more or less immediately, you can identify things which are alive. And the criteria that we use in making this discrimination between the animate and inanimate world has nothing to do with the individual building blocks, such as DNA or proteins. Being alive is a macroscopic state, while things like DNA and proteins are the components of the microscopic mechanisms by which this state is generated and maintained. Let me elaborate on this by talking about this absolutely mesmerizing video that you see on the slide here. This is taken in my group. Now, this is an egg cell or oocyte of a sea star. It's a model system that we use in my group. Note that this is a very large cell. It's about 0.5 millimeter in diameter. What you see is self-organized patterns of signaling proteins that orchestrate a very important process early in development, namely cell division. And of course, the ability to divide is a hallmark of life, at least life as we know it. Now, we cannot see individual proteins. The typical protein sizes are on the order of nanometer. And the majority of us may not even know what they are. But we can all appreciate one thing just looking at this video, and that's the fact that long wavelengths, long structures and patterns that span the entire surface of the cell. They propagate from one side of the cell to the other side. They self-organize from one state to the other state. And this happens while the egg cell is alive. It's continuously consuming fuel and it's dissipating energy. Now, over the past many decades, molecular biology has been very powerful in identifying the building blocks as well as the microscopic mechanisms. However, while we know the list of the parts, it's not enough. And I believe we have made less progress in identifying the parameters that are characteristic of the macroscopic living state. And let me use a quote from Ernest Everett Just to elaborate on this. Ernest Everett Just is a pioneering biologist, where he says that the direct analysis of the state of being alive must never go below the order of organization which characterizes life. Now, if you think about this, this is actually where physics comes in. And to understand the world around us from a physics lens, we need to grasp two fundamental concepts: equilibrium and non-equilibrium. Let's start with the concept of equilibrium, a state of balance and stability. To illustrate this concept, let's consider a simple bar magnet. If we could zoom in, we would see that this magnet is made up of countless tiny domains, each like a compass needle. In an ordinary piece of iron, these tiny compasses point in random directions. But when we apply a magnetic field, something remarkable happens. These domains begin to align. This alignment process is an example of what we call symmetry breaking in physics. Initially, there is no preferred direction for the domains, and this is a symmetry. But when we apply an external magnetic field, this symmetry breaks. The system chooses a specific direction by aligning the domains. And once aligned, these domains reach a state of equilibrium, stable, predictable, unchanging. In fact, if we were to film these domains and play the movie backward, it would almost look identical to the forward version. This is a hallmark of an equilibrium state. Time loses its direction. But this state of equilibrium is not actually where life thrives. In fact, it is the antithesis of life. Life in all its messy, complex, and beautiful glory exists far from equilibrium. Now that we understand what equilibrium looks like, let's shift our focus to where life truly thrives, far from equilibrium. And let's return to our starlings, a much more complex and dynamic example. You can think of each bird in this aerial ballet as a living, flying compass needle. Just as the microscopic domains in our bar magnet align with the magnetic field, these birds align their velocities with their neighbors. But here's a crucial difference. This alignment is ever-changing, responding moment by moment to the movements of neighboring birds. The result is a mesmerizing display of collective behavior that seems to defy explanation. Some people see a thrashing serpent, some a billowing cloud, even a pulsing heart in their formation. The flock moves as one, yet it is composed of thousands of individual decision makers. It's a dynamic living system that never settles into the quiet equilibrium of our bar magnet. Instead, it remains in constant motion, perpetually consuming energy to maintain its intricate dance. This perpetual motion, this ceaseless consumption of energy, is what we mean by non-equilibrium. It's a state far from the quiet stability of our magnetized iron bar, and here is where it truly gets fascinating. It is in this state of non-equilibrium that we find the seeds of life, complexity, and beauty. Now, the starling murmuration is a perfect example of emergence in a non-equilibrium system. Just as the alignment of the tiny magnetic domains in our bar magnet led to the emergence of a global macroscopic magnetic field, the coordination among individual birds leads to the emergence of the flock's complex and fluid-like behavior. But the crucial difference is this. In the magnet, emergence leads to the static equilibrium state. In the flock, emergence is dynamic and ongoing. The flock's behavior continually emerges from the interactions of individual birds, yet it also influences those same individuals, thus creating a feedback loop that maintains this system far from equilibrium. This dynamic emergence is a feature of living systems, and it is the same principle that applies to us all. It what allows for a collection of cells to become a thinking, living human being, or a group of humans to form a complex society. In each case, the behavior of the whole emerges from, yet it also guides the behavior of its parts. So how do we identify and understand these emergent properties? And this is what I'm going to go do today. I'm going to show you how we use experimental tools to observe these non-equilibrium systems. We use data analysis frameworks to identify what are the broken symmetries in our system. And then we develop theoretical frameworks which gives us power of prediction. And then, of course, you can go back in the lab and design an experiment that can verify the predictions of your theory. Now, in equilibrium systems like our starling flock, symmetry breaking is not just a one-time event that leads to a stable state. Instead, it's an ongoing process that's driven by this constant flow of energy and matter. And in fact, we see a continuous cascade of symmetry breaking event. Each break in symmetry opens up new possibilities, new patterns, and new structures. And it's like a never-ending game of dominoes where each fallen piece triggers new and unexpected arrangements. This continuous symmetry breaking in non-equilibrium systems is what allows for this incredible diversity and adaptability of life. And it's why living systems respond to their environments, evolve, and create intrinsically complex structures over time. Now, today I'm going to showcase some of the broken symmetries that we have identified in living systems and the emergent phenomena that we have discovered. So this is a slide that I will walk you through it throughout this talk. Now, we observe non-equilibrium on the grand scale of murmuration. But this state isn't just confined to the macroscopic world. In fact, the principles we discover in our starling ballet are universal, manifesting across all scales of life. So let's zoom in from our murmuration to something much smaller, yet no less wondrous, a single living cell. So each of these is a single living cell. And what you see is within this microscopic world that a drama unfolds that mirrors the dance that we see in the sky above. In my lab, using advanced microscopes, we can watch as proteins, these building blocks of life, organize themselves into waves and spirals of activity that ripple across the surface of a developing cell. And just as each starling responds to its neighbors to create these sweeping patterns of murmuration, these proteins interact with their molecular neighbors to create such coherent patterns that actually guide the development of life itself. And what's truly remarkable is that we can describe such intricate biological patterns using the same language of non-equilibrium physics. And I'm going to show you one example of how we do this. So, for instance, we have discovered that these protein spirals behave like charged particles. And how do we actually discover this? So let's look at the video of one of these spirals. So these are, again, protein patterns that span the entire surface of the cell. And we can do basically data analysis tool. We can use data analysis tools. So for instance, you can see I'm going to look at one pixel on this video, and then I'm going to look at the oscillations of the intensity in this single pixel. And what you end up seeing is actually you can transfer those oscillations and extract the phase of those oscillations from these videos. And all of a sudden, that black and white video is going to become something this colorful. And now you can clearly see that those patterns are spirals and waves that go around the cell. And what's absolutely remarkable is that you can see the core of these spirals, some of them rotating clockwise and some of them rotating counterclockwise. And if you look at the core of these spirals, and you can use mathematical descriptions to assign a charge to the rotating clockwise and counterclockwise cores, what you will see is that they behave like plus and minus charges. And just like electrical charges organizes electric fields, these focal points of protein activity orchestrate the development across the entire surface of these oocytes. But we can do even better. So that's what nature is doing. We can even control when and where these patterns happen. So for instance, we can develop light switchable versions of these proteins so that every time we shine light on our cell, you can generate these patterns wherever you want so that you can dynamically change and control the shape of these cells. And this is an example of how we are controlling these, and we are recruiting these proteins exactly where we shine light on it. These patterns, they never cease to amaze me. In fact, as we watch these proteins dance across the surface of a dividing cell, we witness something even more fundamental, another symmetry that life breaks. And that is the symmetry of time itself. Now, remember our bar magnet. In equilibrium state, time loses its direction. You could play the movie of the magnet forward and backward, and it would look almost identical. But in the non-equilibrium world of living systems, time is actually an arrow. It has a clear, irreversible direction. To appreciate this, let me show you a video of a developing embryo. Here in the first frame of this video, you can see many cells, and to orient ourselves, the cell nuclei is marked in blue. Now I'm going to play this video, and we're going to watch this video for a bit. This is absolutely beautiful development in progress. And I'm sure majority of you are beginning to realize that something is unusual about this video. And you will recognize the fact that actually I'm playing this video backward. In fact, each wing beat of a starling and each cell division in a growing embryo and each heartbeat in your chest is a step forward in time that cannot be undone. Now, here is something absolutely remarkable. We can show that the arrow of time is mathematically related to the flow of energy and to the non-equilibriumness of your system. In other words, time's direction is connected to how energy is dissipated and how far from your equilibrium your system is. We can measure this using a thermodynamic concept termed entropy. Entropy, simply put, is a way to measure disorder. And in fact, if you look at this equation, on one side of this equation, you have an observation of a movie forward or backward. And on the other side of the equation, you have the amount of entropy that's being produced, or which is equivalent to how irreversible the process is or how far from equilibrium your system is. I find this equation absolutely remarkable because it connects something that you can observe to actually something that has a physical meaning and intuition for us. Now, in living systems which are continuously exchanging energy and matter with their environment, higher energy flows means more entropy production and more irreversible processes. And in fact, my group's experiments with these protein waves and patterns have confirmed this. So what you see here is that as the cell consumes more energy, the patterns become more complex and more irreversible. Now, this irreversibility is what shapes how we perceive time. And what's truly amazing is that life can create and maintain complex ordered structures even while it adds to the universe's overall increase in entropy. Now that we have explored these fundamental principles of life from starlings to cells, you might be wondering, how does this change our understanding of the world around us? When we embrace this dynamic view of life, we start asking new questions and exploring new possibilities. How can we use these principles of non-equilibrium physics to create new classes of smarter and more adaptable materials? And could studying energy flows in ecosystems help us discover more sustainable ways to produce and use energy? And if life is really a phenomenon of non-equilibrium physics, should we look for the origins of life in places where energy flows and cycles creates the potential for complexity? And by thinking this way, we open ourselves to discovering new forms of life or lifelike processes that we haven't even imagined yet. And you know the exciting part, we don't even have to look far to see these principles in action. Actually, our own planet is full of incredible diversity that comes from these fundamental principles and processes. Take a coral reef, for example. It's a delicate balance between countless species which are finding their place in the flow of energy and matter. Or think about human brain. Billions of neurons fire in coordinated patterns to create what we call consciousness. Even our societies, with all their complex economic and social structures, can be understood through the lens of non-equilibrium physics. So I'm going to, for the rest of this talk, give you two examples, how we learn from biology to do something new in physics. So how can we design adaptable materials and soft robotics? And in the last part, I will show you how we can use physics to understand some of the most fundamental questions in biology, such as the evolution of multicellularity. Okay, so let's start with what biology can do for physics. And this is an example that was a very serendipitous discovery in my group, so I'm going to walk you through this. And to do this, we're going to start with one of these oocytes that I showed you, and we're going to fertilize this egg. And what you see is that now these eggs are undergoing cell division, so the development has started. This is one of the most amazing videos to watch. It's really biology in action under a microscope. And you can see that you started from symmetric cell divisions that happen many, many times. And in fact, what you don't see is that all those protein patterns that I showed you are responsible for these cell divisions to happen. Now, you can see that at some point we have millions of cells within this fertilization envelope that you can see. And then at some point, you can see that there seems to be a thicker layer on the outside. And these cells are very special. They have a very important organ known as cilia. So they have a hair-like organ coming out of it. In fact, it's the cells that you have in your kidney. And these cells, the cilia, can beat, and it can generate fluid flow around it. And you can see that when it beats asynchronously, it creates this chaotic motion. And when they start beating synchronously, these cells can, these embryos can start swimming around in the ocean. Now, I want to give you a close-up of how these embryos look like. And this is another mode of microscopy that we use. I just want to highlight that you have all these cells at the surface which look like this, which have these hair-like structures coming out, and the whole structure is covered with this. Now, if you want to know how they beat, we have to change again the mode of microscopy. So we're going to look at a high-speed video camera of these cilia beating. And again, what you see is that the surface of the embryo, again, you have millions of cells, and each of these cells, they have this hair structure coming out. And you can clearly see this very interesting wave pattern that they generate. And this is very much reminiscent of metachronal waves that you can get in stadiums. And now, by this type of synchronization, what they do is that they move the fluid flow that's next to the embryo, and that's why they can generate force and torque to be able to swim within the ocean. So how do they swim within the ocean? Let's look at another way of, another mode of microscopy. So what we are going to look at now is looking at the side view of one of these embryos. And what you can see is that they swim around within the bulk. So the bottom part of this video is the bulk, and the top is just a reflection of these embryos in the water interface. What you can see is that they swim around, and then once in a while they come to the water-air interface in a stable configuration. And then what happens is that their swimming motion in the bulk is going to become a type of spinning motion at the water-air interface. So you can think of them as spins at tippy tops, right? That are spinning at the water-air interface, all with the same handedness. Now, what do we do when we have objects like this? Physicists are fascinated by these spinning objects. So what we did is we decided to put lots of them together, and we observed this absolutely amazing phenomena. And they say a picture speaks 1,000 words, and this is a video, so you can do the math. And before I start the video, now we're going to look from the top. And each of these embryos are stable at the water-air interface. And I want you to look at the scales here. So you're looking at something that's a few millimeters across, and also a process that's happening over days. And I'm going to play the video. And what you see is this emergence of incredible collective behavior of these embryos, which thousands of them come to the water-air interface to form what we call a living chiral crystal structure. It's crystalline because they're all the same size. It's chiral because it breaks handedness. It starts rotating in a particular direction, and it's living. And this is not a murmuration. This is really a constellation of sea stars. Now, just to give you an idea of, actually you can observe this with naked eye, this is our attempt on a shot on iPhone. So you can see that basically you have what really looks like a crystallization process that we are used to and familiar. And this is still a living system. It's a collective behavior that's happening over the entire developmental process. Now, let's see why these objects, how these embryos attract each other. So I'm going to look at two of these embryos that are at the water-air interface. And what you can see is that they rotate. I told you that they behave like spins at the water-air interface. And because of this spinning motion, they generate a particular type of fluid flow that causes them to become attractive, and then they attract each other. And then when they come together, they actually have this dance around each other, and they break this, what we call a chiral symmetry, even at the scale of only 2. Now, if you put a lot of them together, again, you have all these forces and that are attraction. They have steric repulsion. They don't pass through each other. But they also have other types of force and torque exchange within these systems that comes from the fact that these are living non-equilibrium systems. So you would not get this from a normal crystal behavior. And because of these additional new types of force and torque that we have in the system, these systems can break a very important symmetry, which is called the action-reaction symmetry. So basically, they violate Newton's third law. And because of that, they exhibit a very interesting material properties that we have discovered. And to let you appreciate this material property, what we are going to do is, you know that the whole crystal rotates in a particular direction. We can do some image analysis to go to the co-rotating frame of the crystal. So this looks like we're sitting on one of these particles within this crystal. And then you can look at the neighbors around you, and what you see is that they synchronously oscillate together. And from an equilibrium perspective, this will never happen, because this type of oscillations in an overdamped in water would quickly damp, and you will never be able to do this, while this system is non-equilibrium and allows you to actually get this type of oscillations, this type of coordination along very, very long distances within this system. We can do a lot of interesting things. We can actually measure the magnitude of these oscillations over the entire experimental videos that we get. But then what we realized was that because these systems are out of equilibrium, because they have continuous injection of energy and consumption of fuel at the smallest scale, they can generate this type of emergent material properties at much larger scale. And this is because they break the usual symmetries that we are used to when we deal with equilibrium systems. And one of them, as I mentioned, is the Newton's third law, this action-reaction symmetry. And because of that, they now have a very unusual elastic properties, which we term odd elasticity, because it's the components in this tensor. This tensor is very much Hooke's law, which is basically just written in this more complex format. And what it means is that we can actually build a material that when you push on it, it rotates. There's no way you can do this when you have an equilibrium type material. Now, what's so special about having this type of oscillations? And we realize that this type of oscillations actually means that you have built an engine from this system. And the way this engine works is that this system is converting the internal energy into mechanical work to offset dissipative losses. And I find this absolutely remarkable, because you're using, again, a rigid body mechanics to build an engine which doesn't require continuous fuel injection. It's basically you kick it once and this autonomous engine is going to perform with efficiency, high efficiency. And do you remember the equation that I showed you, where you can observe your system and measure how much entropy is being produced or how much energy is being dissipated? We can use that same equation on our experiment, and then you can actually measure how much energy is being dissipated within this system. So this heat map is how we use those equations on the data to be able to actually measure this. And you can see that when the system is excited, it appears to be at a hotter temperature. And you can see this basically flow of energy within the system. Now, what can you do with this? Once you have systems that are basically can generate this type of self-sustained elastic deformations, you can start thinking about building new materials out of it. And this is a very fun experiment where my students basically used some printer to create a PDMS shape and then confine the crystal, and then now they have built this wheel of a time reversal asymmetry. But what I want to emphasize here is that this was something we learned from nature. We learned that certain broken symmetries can actually give rise to new material properties. So what we are doing right now is we are using those concepts to actually design robotic swarms. And the video that I'm going to show you right now is basically an example of now a fully man-made example of very much similar to what we observe. So each of these are magnetic disks, and we can actually use a magnetic field, external magnetic field, to start generating this type of swarming behavior. And this is something that I'm very excited. Again, we learn from nature, and we try to emulate that in man-made materials. Okay, so this was an example of how we learn how nature does it, and we build non-equilibrium materials. But I think there is another angle to this, which I'm recently been very excited about, and this has been the focus of the research in my group, and that's the question of what physics can do for biology. And the question that we were very interested in trying to understand is, for the longest time, there has been questions about how the multicellular life evolved and became the multicellular life as we know it. And the idea is that you start from individuals that start interacting with each other, you form a group, and then there is another transition where you get cooperation and robustness within your system, and then finally you have emergence of individuality. And these are the questions that we think we will be able to address with this toolbox of physics that we have at hand. We know very well how we can go from individual agents to a cell aggregate or a group aggregate. But can we also use tools from non-equilibrium physics to understand how the other transitions happens? And this is the questions that we have set out to address. And in particular, we want to know whether we can use this information theory, the equations that I showed you, to be able to come up with metrics that tells us this transition and how this transition happens. So what are the experiments in the lab? We have started with one of the most simplest life forms, sponges. And they are these absolutely remarkable model systems. That's my student Will. And you can take one of these sponges, you can take a piece of that sponge, and you can mechanically dissociate all these cells within the sponge. So you can start from a collection, a soup of cells that are just individuals. And then when you put them together, there was this beautiful experiment from 1907 done by Wilson, where you showed that these cells now come together to form increasingly more complex shapes and structures and finally actually become a full sponge. And this is basically will allow us to really see this evolution of multicellularity in real time. And I'm going to show you the experiments that was absolutely blew my mind when we did it in the lab. So what you see in the first frame is just that solution of dissociated cells. So they're just a soup of cells. And then when we basically added some cofactors and ions within the system, what we observed was this. Now, if you're like me and look at this video, you will see something that very much looks like condensation, right? It looks like phase separation. These millions of cells are coming together, they form droplets, they find each other, and they start coarsening. So this is very much that first step of individual cells finding each other, forming droplets. And if you look very closely, you will see that, so this is a much zoomed in video, you will see how these cells are finding each other, how each cluster starts finding the other cluster. And then at some point, these cells within these structures start making that transition of becoming not just a group, but rather a cooperative group, and then finally becoming a multicellular organism. And this is what we will be monitoring and trying to understand from the lens of physics. Now, okay, I think this is time for our journey to come to an end. And for our journey to come to an end, I want to revisit the starlings again, now seen through new eyes, of course. As the last light fades, of course, they're bringing their ballet to the final curtain, settling gracefully into their roosts. Through them, we saw how energy drives matter to self-organize, how breaking symmetry can create complexity, and how arrow of time emerges from life's processes. I will never forget that moment in my lab, watching through the microscope as protein waves rippled across the surface of the cell with energy flowing and driving this ballet through time. That moment fundamentally shifted my perspective. I realized that life isn't about reaching equilibrium, but about sustaining this beautiful dance far from it. And it is in this constant flux, this journey of becoming, that the true wonder of existence lies. A dance we are all part of every moment. And I want to end this by reciting one of my favorite poems that's tributed to Abu Ali Sina, an Iranian polymath. دل گرچه در این بادیه بسیار شتافت، یک موی ندانست ولی موی شکافت. اندر دل من هزار خورشید بتافت، آخر به کمال ذره‌ای راه نیافت. I'm going to leave this you with references if you want to learn more about the details of the work that I do. The reference on the right will be helpful, and also I'll invite you to read an interview that I did to learn again more about the details of the work. And I'm going to just take a few more minutes. This is my incredible group at MIT. This is, of course, the work of a group of undergraduate students, graduate students, postdocs, and of course my collaborators across the globe. Some of my alumni are now here at Stanford. And at the end, I want to also express my gratitude to my parents, who has always been this boundless source of inspiration and unrelentless energy for me and my sister, Bita, who's a faculty member here at Stanford. And I'm really grateful that they could be here today in the audience. And then lastly, this talk is to all women, to women of Iranian heritage, including Christine Mona Khademi, who have pushed and continue to push the boundaries every day. Thank you. And I would love to get questions. Yes, please.
Thank you so much for this informative presentation. I enjoyed it very much. I didn't understand a lot of it, but I have two questions. The first one is that your first slide that you showed an egg cell and the proteins congregating and propagating. I was wondering if you had a living egg cell and a dead egg cell. The difference is that on the living one, the proteins propagate and the other one not. Is that, am I right or wrong?
Yeah, no, absolutely right. So the dead cell, you wouldn't get patterns like this. But I'm going to actually rephrase your question in another way. So I could have showed you this video, and if you are a chemist, you would have told me that they can actually make a reaction diffusion pattern like this in a test tube. So what is the difference between that and my living system? And the answer I will give you is that these patterns, they have purpose, they have a function. And I think that is one of the most important signatures of living systems. Patterns evolve depending on the type of function that's required from them. And in fact, you see that these patterns, they seem elongated. And the really absolutely, I guess, fascinating thing to me is that if you think about the size of the cell, this is a 0.5 millimeter size cell, and then you have proteins that are only nanometer size. So they wouldn't know at all where they are within this incredible volume and size of the cell. And it is this cooperativity that allows them to actually be able to function over much longer distances and scales that are just otherwise impossible to do.
My second question is that if you go all the way back to the Big Bang and the creation of the universe, can you say that the creation of matter and the cosmos was because of symmetry breaking? Right?
Absolutely. That's actually one of the most fundamental areas that they think about, the symmetries and the broken symmetries and how
They talk about matter and antimatter.
Absolutely, right. Yes.
Fluctuations that bring these, and then there's a little bit more matter than antimatter. That's why we are generated.
Absolutely.
The final question is that, are you, your group, working also in the origin of life research that was done in the 50s in the University of Chicago, Miller and his group?
So we're not working on the origins of life. I think my current interest is understanding one of the major evolutionary transitions, which is the origin of multicellularity. So this comes much later. How do you go from a single cell to a collection of cells again to becoming an organism and then dividing, division of labor and everything. So that is the area that I'm going to focus. But I think the origins of life is also an absolutely incredible area of inquiry, and yes, maybe at some point. Yes. Maybe I'll start there and then.
Okay, first of all, I absolutely loved your talk. It was so beautiful, but so complex. And throughout the talk, I try to connect it to my daily life. So I hope you can maybe shape this in a more proper way. But from what I understood, in our daily life, things can get chaotic and out of motion. And that's not necessarily a bad thing. Instead, we should find where there is a way to make it flow, make it dance together. And then also, I remember you said time, it's straight, it's linear, it's actually an arrow or moving away from equilibrium, yes. Yes. So I'm understanding this correctly?
I think that's a beautiful interpretation of what I said. I think at the end I tried to say that life is really being away from equilibrium, embracing this non-equilibriumness. In fact, we say that when you reach equilibrium, you're dead, right? And I think this constant motion, I think it's this perpetual motion, is something that I think about in my daily life as well. And I think that I think was a very beautiful interpretation of my talk. Thank you. Yes.
So one of the images you shared earlier were the patterns. Like, were there a confined set of the number, like a limited number of certain patterns that you observed, and everything was basically like set up a complexity over those limited sets?
Yeah, that's a very interesting question. So we can actually do this numerically. So we can write down equations and then map the whole phase space of what are the possible patterns. Experimentally, you have access to a set of these patterns. And that actually, to me, is a very interesting question because it means that biology has some constraints and picks a particular part of the phase space of all possible patterns. So experiments, basically what we do is we systematically change the amount of fuel that's available to the cell, and that's kind of like you can tune the complexity of the patterns.
So in other words, are there like building blocks per se of these sort of patterns, and then like have you mapped those out in terms of like there are 5, 6 number of like set patterns, and then it kind of functions as say like base pairs and like DNAs or where there's 4, but is that kind of how you see it?
I think we're looking at a very different scale. So in fact, it's a scale where we call more is different. So you actually can have a continuous phase space, and just changing parameters a little bit, you can end up in very different directions. So that's I think how I think about it. Yeah. Sure. Yes.
Thank you so much for your talk. It was really interesting. I think the part that was really interesting to me was the part that you talked about that living mechanism with a little bit of energy, that non-equilibrium can go on forever. And then the part that you were saying that like with getting inspiration from that when you showed that swarm robotic example, trying to mimic kind of that behavior in materials. I guess my question is, why can't we use the actual biomaterial as the material instead of like mimicking them and trying to like, definite the behavior? Because I think your students were doing that like we're trying to like, I guess contain it in a ring. Mm-hmm. Why can't we use that as material?
I think that's a very interesting question, and a lot of people are interested in this area of biomimetics, so combining living and non-living materials to form these hybrids. And I think you can perfectly do that. The question is whether you can have control on biological systems, but oftentimes you can have more control on systems that you build from bottom up. So for instance, we can, if depending on what type of functions you want from that system, for instance, robotic swarms, it may be that it's much easier to control something that you know exactly how it will behave, like depending on the parameters that you give to the system. But I think that's obviously a very interesting question. Questions you can ask is actually why would biological systems do this? And if we understand what the function of these type of emergent phenomena is, then I think that's another way to actually think about your question. Yeah, thank you. Sorry, I have seen your hand in here.
I raised my hand so many times.
Yeah.
Beautiful talk, thank you so much. I guess the first question is, what motivated you or inspired you to go into this field and this specific research? And the second part of it is, what are the biggest obstacles that you're facing in trying to get to the big questions you're trying to answer? So just give us a picture of what that looks like.
Yeah, so I started as an engineer, But I would say that engineering is about optimization, but I always wanted to understand how the system does it and going beyond just optimizing parameters to get like a particular function. And I think over my course of, or the trajectory that I took coming from Iran as an engineer, I started becoming more and more interested in pursuing this type of physics questions. And initially I started thinking about materials and soft matter. And soft matter is, I'm a chemical engineer by training, and soft matter is a very easy extension of chemical engineering. But then towards the end of my PhD, I realized that there is this whole world of biological systems. And the way I look at biological systems is they can be a playground for doing new types of physics, new types of soft matter, this non-equilibrium matter that can open up a whole range of possibilities. And I think that's when I realized I saw the light and I kind of shifted in that direction. I would say that as I'm kind of getting deeper and deeper into this, I think to me it's really like an Alice in Wonderland every day. You go in the lab and there's always this new discovery, and there's a lot of kind of downs as well, but there's those moments that you see something under the microscope, and then it's just you want to know why. And I think that always has been my motivation in trying to kind of understand like the phenomena that we observe. Very recently, I think with the tools that we have developed, I realized that we can actually also start asking more fundamental biology questions, and I think physics can be very useful in trying to actually bridge the gap between how biologists think about their systems. And that's the thing that I'm very excited about and will be pursuing. Yes. Yes.
Very nice to see you.
Samira.
Great talk. I really enjoyed it. I'm a little bit more proud. So one question that I was thinking about.
Samira is my dear friend from Sharif.
Yeah, I think I saw her picture after the picture that you had.
Yes.
Really honored. So I was wondering, this beautiful talk that you talked about, this dance of non-equilibrium. Have you ever thought about like natural aging process, for example? What that do to this like non-equilibrium state? Or in extreme cases, things like cancer, which might be more complicated, but that's just aging.
Yes. I think that is absolutely a very timely question. And I think those are the questions that usually you try to address this using biology, using genetics and all these things. But I think this energetics, I think this is one of the kind of like most maybe overlooked areas. And I think the question you're asking can be really like basically framed in this, I guess, in this framework that, like if equilibrium is dead, then maybe you can start thinking about like the energetics of your system, and then how maybe you can frame aging in that kind of like context. But cancer cells is even more interesting. When we started thinking about kind of this origins of multicellularity, how you get cells that come together, how can you kind of quantify this cooperativity? And I think you can maybe rephrase that as basically cancer being a state where the cells are no longer cooperative and they kind of start migrating to other. And I think to me that's just a whole new way of thinking about this system. So yeah, thanks for that question. I think this is a very exciting area to explore. Yeah, very nice to see you. Yes, sorry. Josh, my wonderful undergraduate who's a graduate student here.
Thank you for your talk. So for the sponge coarsening, how is the domain size scaled? Is it well described by convolution?
No. So we have looked at the scaling, so we actually don't see that scaling, and we're trying to basically see whether we can, whether you need to modify those equations. So it's not a simple convolular. Yes, yeah. That's great seeing you. Yes.
I'm just wondering, how do biological organisms like strike the balance between like order and then this asymmetry and this kind of chaos? Is there like a sweet spot that like fosters life?
In fact, energy dissipation is paying the price to be ordered. right? So biological systems need order, and they use this energy dissipation to create order within them. So it's a price that they pay to be able to keep order. Yes. So maybe I will also say that it's a delicate balance as well, right? And I think that's kind of like one of the most remarkable things, that many things can go wrong. We're really living in this very narrow region, but then somehow they can do this so well. And I think that's another thing to try to learn. Yes.
Hi, thank you. I was really fascinated by this talk. I'm currently a Stanford undergrad studying bioengineering, so this is really fascinating to me. And in a similar vein, actually, to that question, this idea of non-equilibrium was really fascinating to me. And it was also discussed in the presentation that as molecules, a very famous law in thermodynamics is that we strive to tend to this high entropy state of disorder. But would you say that disorder is what causes the balance and the equilibrium on the macro scale, or what is like that dance between the two?
I think maybe kind of like going back to the previous question, I think basically the entropy is always increasing, right? But then you need to pay a price to do that. The entropy is disorder, and you need to pay a price to keep order. And I think that dissipation is basically that the price you pay to be able to keep the system because order means function. Order means you have a particular structure that will perform a particular function. And I think that's so there is no violation of second law. It's really just how your system continuously dissipates energy and comes up with mechanisms to be able to maintain that ordered structure. I think that answers it. Yes?
So have you looked at the smaller scale amino acids? Because we have this asymmetry in amino acids in living systems. They all have the left-handed amino acids. Mm-hmm. Any comment on that? And the second question, the spinnings, the videos you showed, they are mostly, I think, all of them are clockwise. Is it because we are in the northern hemisphere?
Yes. Question that I'm, so maybe first talking about the chirality. So what I will comment is that the chirality you're talking about is chirality in structure. So DNA is chiral. The chirality I talked about is chirality in dynamics. So they have a certain dynamics. They break left-right symmetry, handedness in that kind of scale. And it's interesting, you may ask, why do they break that kind of symmetry? In fact, it's the same signaling pathway that gives rise to left-right asymmetry in many organisms, including us. So that's the same kind of like cascade of signals. And so that's, I guess, the mechanism for breaking this type of chiral symmetry. Now, they will not rotate in a different direction in the southern hemisphere, though they actually do respond to their environment. So oftentimes we see that when they are within this kind of crystal state, depending on the presence of strain or strain fluctuations around them, depending on how their neighbors behave, oftentimes you can actually get a distribution of frequencies, but also handedness. And I think that's a very interesting question we're trying to address. The way we think about it is that you can have a double well potential. The majority wants to rotate in one direction. Once in a while, depending on their interaction with their environment and the cue that they get from their environment, they rotate in the opposite direction. Yes. There and yes.
Yeah. Thank you very much. My question is probably the odd one out. It's really interesting that you are making a relationship between this complex concept and life not only as a biological concept, but also as a cultural, social concept. So my question is, to what extent do you think we can use this framework to really find some of the social phenomena like inequality?
I think these are really interesting questions, and there are actually many scientists who think about this. This idea of basically how very local interactions. Your local environment can give rise to collective behavior or like a collective phenomenon on a much larger scale. This is basically at the gist of this. And I think there are many ways to think about this in many areas. Even thinking about large language models and how you get emergent phenomena there, that could also be something that you can use tools from physics to think about that. So I think that really is open to really be able to think about social context, economic context, and any other. Yes. Thank you. There was a question there.
When within nature the system become in a state of order, then after it becomes in a state of order, then it starts become to the state of becoming. Is it switching back, going order, becoming order? Because is that what's happening in the nature?
I think in nature, you can have many types of order. And I think what I tried to maybe answer one of the questions was that in nature, order and function are the two sides of the same coin. You have a particular, for instance, ordered structure because there is a particular function that needs to be fulfilled. And I think these are the two sides of the coin. So going from order to disorder and back to order is really depends on what state your system is and what kind of like how is it trying to kind of, I guess, fulfill a particular function.
All the time it cannot stay in the a state of order. All the time it cannot stay in the state of order.
It can, I think, but I think what's important is the fact that they interact with their environments, and sometimes they get a cue from the environment so that they now need to change their state of order and move to another state. Yes.
Have you ever tried combining a collection of cells from more than one organism, or from the same organism at two different developmental stages?
Yes, absolutely. I don't have a video of that. So we did 2 developmental time points. And you can actually tune a very interesting parameter there because the way you can tune their interaction parameter. And you can get actually a very interesting state. So here, only when you have basically a pure particle, pure developmental stage, you get a crystal. When you mix them at 2 developmental time, you actually can get flocking also. So you can get a flocking state and then a crystal state. So there is a lot of, you can play these games. We are very excited to try that with sponge because you can have sponge of different species of sponge. And one of the questions we want to ask is whether you can identify who your this likeness and whether you can actually get some interesting information there. Yes, thanks. Yes.
Have you considered any other like diseases or anything that may be impacted due to these like clustering behaviors, like protein-based, like Alzheimer's or anything like that?
It is not my direct area, but I would say that I actually had a wonderful graduate student. So this is looking at protein condensation. It's a whole area. Many, I guess, important work is being done in that area. We actually did a little bit of that. My graduate student is now a postdoc here, in fact, at Stanford. And we tried to create an in vitro example of that. So we basically took proteins out of organism and try to see whether you can get this type of condensation. And we try to actually see the effect of ATP, which is the fuel, on these cluster formation and the material properties of the cluster formation. We have a couple of publications on that, and there's actually a lot of interesting questions to think about whether you need non-equilibriumness, you need this basically ATP consumption to keep the cells fluid, or these, and whether lack of that can actually lead to like this type of, I looked at it from a physics lens, but I think there are many people who are thinking about this more in terms of disease and trying to address that. Yes, yeah. Yes.
Hello, thank you so much for your talk, by the way. I had a question with respect to the experiments about the sponge. It seemed as though the focus of that experiment was to try to identify how single-cellular organisms evolve into multicellular organisms. And it seems like a prerequisite for that question, or for understanding or trying to answer that question, is trying to identify a point at which the collection of cells becomes a cooperative of cells, as you worded it. And so I'm curious as to how you can characterize basically, yeah, the inception of multicellular life. Like, how do you characterize it biologically? How do you characterize it physically? And another question related to that.
Yeah.
I guess this is a question about the structure of the sponge as a multicellular life form, but if they're anything like humans, they're different, like their cells are differentiated. And so how is it that these differentiated cells, like assuming they are differentiated, maybe they're not, but like does that also complicate the way in which you kind of see these collections versus cooperations?
You should have come to my group meeting last week. This is a very kind of early development in my group. So I would say that the experiments are maybe a few months old, and we're really trying to actually answer those questions. So I'm going to kind of tell you how we're thinking about it. Right now we're collecting data. But we think that maybe signature, so at the very early stage, as you mentioned, is basically whether we can kind of see how this transition of, okay, we can see we have cells, they clump together. That I think is a phase transition, although it's not clear if this really follows the kind of the scaling laws that you get from traditional phase transition paradigm. But then the question is whether what would be the next stage? Like how do we then identify basically this transition from kind of like a clump of cells to something that's now more cooperative? We have ideas, like we think that it has something to do with the scale of information. How does information changes? Whether you start basically kind of like developing chemical ways to communicate, mechanical ways to communicate. So we think that if we can actually look at the onset of those kind of emerging features, then that could be a way to think about it. So this is really just hypothesizing at the moment, but I really think that the way I want to kind of imagine this is basically we can have like this information transmission and scale as a way of trying to quantify this. Now you brought another interesting question, which is are these cells differentiated? Actually, they're quite marvelous. So there's only 3 cell types within this structure, I guess plus and minus. And then they have this tendency to actually go back and forth. So they can become stem cell, migrate, go another place, differentiate again. So there is this absolutely interesting dynamics that happens also on the scales that you're observing the system. So when Will came to my lab and kind of introduced me to this system, I feel like this is just there's so many interesting questions to try to answer, and whether like it's not just genetics, but maybe mechanical cues or this type of information again can also like basically give you some hints towards again like differentiation and other. So it's a very interesting question. Yeah. Yes.
Do the cells have memory, and where do they get their energy? And as you just said a second ago, they move around.
Yes.
Is it dependently decided that this is where we're going to go?
We don't know. I mean, these are very interesting questions, and we don't know whether. And to do that, usually you have very standard model systems where the genome is mapped and all these things. I think these are less studied systems. So we're now trying to put in place all the tools we need to be able to actually also look at the expression patterns and everything, whether to see. Do you have memory, or are there kind of time scales of memory? I think these are very interesting questions. I don't have answers, but maybe in a few years, I feel like I'm so excited. This is the area that I want to explore for the next few years, and I hopefully maybe in a few years I can answer some of these stuff. Yes?
Just a, do you ever see a divine influence in this whole area of research?
I believe there is something bigger than us. Yes.
Can you say a little more about when from the individual cell you go to a collection of cells, and how then those cells evolve? Were you just referring to that a moment earlier?
Right.
That's an area of new research or next research?
Right, yeah. So maybe I will bring this back because this is actually a diagram that we made last week. So this is actually really the question we're trying to understand, which is at the single cell level, maybe there is some kind of chemical field around you, so which allows you to transmit information very locally so you can find neighbors. And that will then lead to formation of cell aggregate. But a cell aggregate is really not a cooperative aggregate, or we want to characterize it as just being a cell aggregate, but not stable. And then when you have other mechanisms of transmitting information, so you learn that you're actually a part of a collective, you start developing cooperativity. And cooperativity, then we want to basically kind of, I guess, characterize cooperativity as the scale of information processing that you kind of like, I guess, develop for communicating, or modes of communication that you start developing. And then eventually you basically start developing polarity, you break other types of symmetries, and that then you can quantify or identify that as a multicellular organism.
In preparation for your talk, I read The Song of the Cell. And she talks about how within a cell you have different areas developed that have different functions.
Right.
And then when you get to multicellular organisms, those functions are taken on by specific groups of cells. So that's
I think that is something that we are definitely interested in trying to explore. In fact, with sponge is a really interesting model system because you have it's a filter feeder. So you have 3 cell lines that does 3 things. Like one is the structure, one is basically cilia that can feeds the water. So these locally, depending on where you are, you have this kind of like types of cells that then give rise to the function. So it's very much like within a cell, and then of course within an organism. Yes. Thank you. Yes.
I had a question actually about the slide, and I'm wondering about something you spoke about earlier. So you can see some patterns in non-living systems that are similar, and that at some point there's a lot, there must be some intentionality behind the patterns. So at what point within this gradual growth towards the multicellular organism do you see a need for intentionality, and is there any place in the math for that?
Yeah, I think that's a very interesting question. Again, I would say that this is very new area, so that I'm just putting our how we want to think about this. But in fact, like this kind of, I guess we want to quantify this intentionality or individuality or some kind of cooperation or robustness. I mean, these are, I guess, terms that like we want to be able to find a connection between information thermodynamics and then use those metrics to actually look at the system and say, okay, this is where the transition happened, for instance, whether it was a sharp transition or a gradual transition. So I have ideas how to move that, but first we need experiments, and I think we're getting really good experiments, but then kind of thinking hard about how do we quantify that. I think this is a challenge that I'm very excited about to address. But yeah, thanks for the questions. Yes.
I have 2 questions actually. The first one, I know that this idea of life and just such a process of hosting life is hand in hand with this disorder. When you pass on, what is the process of that disorder? Do all cells die at the same time? Is there a certain kind of equilibrium that is cumulatively reached? Have you done any research on that?
I have not, but I think that's a very interesting question. My sister is a medical doctor. I always say one of us is saving the world, and I think she's that person. But I think those are very interesting questions. I don't have answers for those, but I can. Yeah.
Maybe I'll do research. My second question, unrelated, is this idea of murmuration was really interesting, and these flocks being able to have this like-minded, sense of direction and time. Is there a way that research is proving that AI technology can replicate that sense of nature, maybe even one day better than nature itself? Is there any research that's been done to try to mimic these patterns that we see inherently?
As a matter of fact, this idea of how flocks behave came from animation. People were trying to kind of like come up with ways of doing. In fact, one of the Batman movies where you get like these bats coming in, this was like the idea that you have agent-based models and you put very simple rules in your model. So always that the rule that you put is I'm going to look at my neighbors and I'm going to do exactly what they do. And that is enough to actually generate these patterns over very long scales. And that kickstarts my field, which is called active matter.
which is basically now can I understand all the different classes of systems and how basically local rules can give rise to this collective behavior. So, and I think for the AI, I think a lot of people think about how, I mean, large language models, they also have very interesting emergent properties. And there are people who think about how these properties emerge because we also have interesting local interactions. And I think this is, again, like it's a science that has, I guess, yeah, I mean, it could be probably applied to many interesting areas.
So I'm fascinated by this picture because every multicellular organism I can think of, plant, animal, starts from a fertilized egg or a single cell, maybe mitotic or meiotic. And so then signaling with various developmental signals develop into differentiation in multicellular organisms. Is there any naturally occurring multicellular organism that aggregates first a bunch of self and then reorganizes?
Yes, sponge. Sponge is this, these are the 1907 experiments done by Wilson. So he took a piece of sponge, he basically mechanically sheared, or he used calcium-free, magnesium-free seawater. All the cells got dissociated. They got de-differentiated to their stem kind of state. And then he puts them together, and basically he sees the experiments that I showed, which is basically they come together, they form first clumps, then they actually start developing the whole sponge. So, and sponge is really at the bottom of kind of like the multicellular life. So I think this is to me, it's like thinking about what is the hydrogen atom that I can start kind of like exploring, and then of course you can get more complexity, and there were many other ways of, I guess, evolution. Choosing different pathways to be able to.
So sponge is the only one.
I guess I don't know,
okay.
but at least sponge, I know that this is what it does, right? Yeah.
Thank you so much.
Thank you.